Hello everyone. Sorry for the inconvenience. So actually, this talk is also kind of a special for me because I was the previous student from the Giriraj Government College. I'm always thankful to my teachers. Alim Khan sir also was my teacher, who taught me botany, biotechnology, and microbiology. So with this, I'd like to start my topic. So basically, the topic for the today is molecular tools that drive scientific research and its basic principles and application of plotting techniques. So if you see, somehow I'm not able to move the. Okay. Yeah. So what are the molecular tools that drive scientific research? So basically, there are some blotting techniques. These forms basics that drives all the scientific research. So without these techniques, any molecular biology laboratories that are working, it won't work properly. So basically, these are the techniques that are essential to drive all the scientific research. So what exactly are the blotting techniques? So what is the definition of blotting? So blotting is defined as a, in molecular biology terms, it defined as a transferring of DNA, RNA, or proteins from an electrophoretic gel to a carrier. Then, after transferring the DNA, RNA, or protein to the carrier, that is nitrocellulose or a PVDF membrane, and that membrane is subjected to further visualization techniques via hybridization of nucleic acids and chemiluminescence or some other methods which are used to develop for the visualization. So, what is the application of this blotting techniques? So how exactly this is applicable? This blotting techniques. So as you can see, I can give you a best example. So if you want to find a hay, if you want to find a needle in the haystack, so what you will do? What you do is basically either you use a magnifying lens or you use some kind of magnet, right? So in order to find the needle. So in the same way, so our cell also has different proteins, DNA and RNA. Let's say suppose example. So if you wanted to find the specific DNA fragment, or if you wanted to find the specific RNA, or if you wanted to find the specific protein of interest in a cell or in a mixture of a protein, DNA and RNA, so how do you find it? So basically blotting is a technique so which is used to visualize a specific fragment of DNA, RNA or protein. So depending upon the type of blotting, so it's basically divided into the three types. So first one is southern blotting, and it is used to detect the DNA. And the second one is northern blotting, it is used to detect the RNA. And the western blotting, it is basically used to detect the protein. So these are the three different types of blotting. So first we'll go with the southern blotting. So southern blotting is a technique which is developed by Edward Southern in 1925, and as I have mentioned earlier, so it is basically used to find or detect the DNA fragments. Basically, this involves three steps. First step is digestion of DNA by restriction endonucleases and separation by Agarose Gel Electrophoresis. And then second step involves the transfer of DNA fragments onto the nitrocellulose or a nylon membrane. And the third step is followed by hybridization with a probe, and then followed by a detection. So we'll go through the different steps now. So first step, as I have mentioned earlier, so first step is the digestion of DNA fragments. So first step, what you do is basically you'll take the DNA of your interest, so where you want to find the specific DNA fragment, and then you digest the DNA with the different restriction endonucleases. So what are restriction endonucleases? Restriction endonucleases or the enzymes or kind of a proteins. Basically what they do is basically it will cleave the DNA at specific sites. As you can see here, this is a double stranded DNA, and this EcoR1 is a restriction enzyme. Basically it cleaves in between G and A. So basically the DNA is very large, right? So if you want to detect a particular portion of the DNA, basically you have to digest it with the different enzymes. So those are called as restriction endonucleases. So then once the digestion is done, so then you proceed to the next step. So the next step is agarose gel electrophoresis. So what is agarose gel electrophoresis? So agarose by name itself is indicates that basically agarose is a polysaccharide and it's a derivative of agar, and this agar is basically isolated from or kind of purified from the algae, and this agarose is basically used for separation of the DNA. So, and this main principle of this agarose gel electrophoresis is it separates the DNA based upon the charge. DNA is a negatively charged. The charge of the DNA is negative because, as we all know, it consists of the phosphate group. So, because of that, it will have a negative group. So, under the constant electric current, the DNA molecules, the DNA fragments will get separated and they move toward the positive charge. So as a result, the DNA fragments will be separated on the agarose gel. And basically, the separation mainly depends upon the agarose concentration. So agarose concentration, depending upon the agarose concentration, the pore size of the gel is maintained. So basically, if you use less agarose, then more pore size, and then more agarose, less pore size. So basically, depending upon this, the pore size is maintained, and also depending upon the molecular size or molecular weight or molecular size of the DNA fragment, the DNA fragments will get separated, and also based on the molecular conformation. So this is how. So the first step is basically fragmentation of the DNA, and then second step is the separation of the different fragments by using the agarose gel electrophoresis. Then we proceed to the next step. So basically, it is visualization. So as we all know, basically whatever you deal with, it's all invisible. Basically, DNA, RNA, or protein, whatever you see, it's basically watery, right? So you, if you want to visualize the DNA and RNA, so you need some kind of a chemical or something which imparts some kind of a seeable nature to the DNA or RNA or protein. So in this DNA, so for DNA, what we use is ethidium bromide. So ethidium bromide is basically a chemical. What it does is basically it acts as an intercalating agent. So it basically it stacks between the two, between the double-stranded DNA, and it binds to the DNA. It basically intercalates between the two DNA strands, and then once you illuminate that with the UV light source, so you can see, you can visualize the DNA fragments. That's how you visualize this DNA fragments. And basically this ethidium bromide is a carcinogen, so we should be very careful while using this ethidium bromide. And so that's why the new dyes have been developed. So these are called as cyber dyes. These are highly sensitive and less toxic. So now we have seen the DNA. So we have confirmed the DNA fragments by using the ethidium bromide, that is UV source, by using the UV source. Then we proceed to the next step. So in this step, what we do is basically, this is a blotting. So the blotting is a method where you transfer the DNA fragments in the gel to the membrane, onto the membrane. So basically the membrane that we use for DNA is a nitrocellulose membrane or a nylon membrane. So basically nitrocellulose membrane has a pore size. Basically it has a small pores, and due to the hydrophobic interaction, so what happens is that the DNA which is present in the agarose gel will go and bind to that nitrocellulose membrane. So for that, basically what you have to do is, the DNA which is present in the agarose gel is double stranded. So you wanted to make the double stranded DNA into the single stranded form. So in order to make the double stranded DNA into the single stranded form, you treat that with the sodium hydroxide. Basically you are treating it with the alkali agent. So as a result of which, the hydrogen bonds between the DNAs will get broken up. Between the two DNA strands will get broken, as a result of which it will form the single stranded DNA, two separate single stranded DNAs. So that is a step called as a denaturation step. And this single strands binds more efficiently to membrane. So then, so basically buffer is used to facilitate this transfer. And then basically this transfer of DNA fragments to the membrane basically relies on the capillary action. As I have mentioned earlier also, it basically hydrophobic interactions are involved in the binding of the DNA to the nitrocellulose membrane. And in some cases, or vacuum or pressure, so different methods are used to aid this transfer. The faster and most efficient transfer is also achieved by means of electroblotter. So basically what you do is, basically you supply the electric current, as a result of which due to the capillary action, due to the electric current. So from negative to positive charge, this DNA will move, as a result of which the DNA will get transferred to the membrane. So this electroblotting depends upon, depending upon the fragment you wanted to transfer, it might take from 1 hour to 4 hours, in between 1 hour to 4 hours. So, and the next step after transferring is basically the hybridization. So the hybridization is a technique where you use a specifically radioactive labeled probe with the DNA fragment of your interest. So let's say if you wanted to detect a X fragment in the DNA, so you basically, what you do is you make the probe radio labeled to that specific fragment. And once that fragment are made, radio labeled fragments are made, basically what you do is you incubate that radio labeled probe with the your membrane, and then after that you'll do the washing steps to remove the unbound probes, and then followed by autoradiography. So what, during the autoradiography, what happens is, as you are using the radioactive labeled probe, it will emit the light, as a result of which it will get transferred to the X-ray film, and there you can visualize the DNA fragment of your interest. So this is how the southern blotting works. So basically, what you do is you'll take the DNA, or you isolate the DNA from the source. The source can be either the plant cell, or the that is plant cell, eukaryotic cell, or any cells. So the source can be anything. So where you wanted to look for a specific fragment of DNA, and then after that, what you do is you isolate. After isolating, you'll fragment that DNA by using the restriction endonucleases, and then followed by the fragment separated by Agarose Gel Electrophoresis, and then followed by the transfer. This is a blotting method. So in this blotting method, what you do is basically you use a filter paper and a gel, and on top of it, basically you place the membrane and again the stock of filter papers. And then in this case, you can put the weight, or in kind of electroblotting, you use the electric current. So what happens is, from down to up, some from negative to positive, the current will move, as a result of which the DNA fragments will get transferred, or kind of blotted onto the membrane. So onto the membrane, basically these DNA fragments are blotted, and then once this blotted membrane is there, and then you basically you do the denaturation here, and then followed by adding the probe here, and then you do the autoradiography, and then you visualize it by using the autoradiography by using the X-ray film. So this is how the southern blotting works. And then we go to the north. Then what are these applications of this southern blotting? So basically, southern blotting is used for gene discovery, or it can be used for a mapping of the genome. So, or in order for the evolutionary studies, so it can be used, or it can be used for diagnostic and forensic studies. Let's say if you wanted to find a specific culprit, so you can use this technique to find the specific culprit, because every humans have a different DNA fragments, right? So depending upon that, so you can use this technique to find the culprit. So basically, this is a technique what they use in forensic studies. And then either you can study the deletions or insertions, or you can also study the point mutations. So if you want to know particular point mutations which occurred in the genome, then you can find it by using this southern blotting technique, or you can also apply this for using these structural rearrangements, or a presence of a particular bit of DNA in the sample. So these are the basic applications of the southern blotting. So then next we will go for the northern blotting. So northern blotting is also technique. Basically, it's used for transferring the RNA sequences, or kind of detecting the RNA sequences. This method is developed by James Alvine and James Stark. So RNA molecules have a much defined length, because DNA is very large, right? And RNA molecules are basically DNA is transcribed into the RNA, and these are of a specific size. So these are kind of a much smaller compared to that of the DNA. And then these RNA fragments are separated by using the same procedure, like gel electrophoresis. And then, so basic thing, what you have to care here is RNA is more susceptible for degradation. So you have to be very careful while isolating the RNA or doing the northern blotting technique. So always you have to do it in the cold conditions, and you have to wear the gloves, and you do not expose that RNA to your bare hands, or anything bare surfaces, so that the RNA get degraded. and then RNA is also blotted onto the positively charged nylon membrane, and it's the same procedure. Basically, here you don't have to degrade the RNA with the restriction endonucleases because these are already smaller fragments, so you don't have to degrade it with the restriction endonucleases. And then after that, basically what you do is you do the gel electrophoresis, and then after that you transfer that RNA onto the membrane, and then you detect it by the radiography. So this is how the northern blot works. So basically what you do is basically you take the sample of your interest, and then you isolate the RNA, and then after that you do the electrophoresis. So this is how you separate the RNA depending upon the size, and then after once this the RNA is separated, so then you transfer that RNA onto the nylon membrane, transfer onto the nylon membrane, as you can see here, and then you add the radio labeled probes. So which where these probes are exactly designed to the fragment what you want to know. So basically, these probes are basically designed against the fragment of your interest, and these are radioactively labeled. So that's why you can use the autoradiography. So basically, it involves the using of the X-ray film. So it has the radioactively labeled probes. So what happens is wherever this radioactive probe is bound, that area will get exposed to the X-ray, and it will emit the light. And as you can see, you can visualize it as this kind of a black colored bands on the X-ray film. So this is how you detect whether the a particular fragment of your interest is present in that sample or not. Then we go to the Western blotting. So basically, sorry, application. So what are the applications of this northern blotting? So this is a standard technique basically which is used to study the gene expression at different levels, and it is also used for detection of the mRNA transcript size, or study of RNA splicing, or study of RNA half-life. These are the different some of the different applications of northern blotting. So then we go for the Western blotting. So Western blotting is a technique. Basically, it is used to detect the protein of interest from the mixture of a protein. So basically, if you want to detect a particular protein of interest in the mixture of proteins, so what you do is you basically use a Western blot. And this technique was developed by George Stark, Harry Tobin, and the name Western blot is introduced by Neil Burnett. So basic principle of a Western blot relies on the molecule of interest, that is a protein, and if you wanted to kind of know a particular protein of your interest is present in the mixture of many other similar molecules, this technique is used. And molecule of interest is protein here, and the probe used basically in kind of a DNA and RNA in kind of a southern blotting and northern blotting, you use the radioactive labeled probes, DNA probes, or complementary DNA probes, or you use the RNA probes. But in this case, in order to detect the protein, what you do is you basically use the antibodies. So what are antibodies? Antibodies are basically proteins raised against the protein of your interest, and these are basically used to detect the protein of your interest. So how exactly this Western blotting works? So Western blotting works in a different steps. So first step is tissue preparation, second is SDS page electrophoresis, and third is transfer of that protein onto the PVDF membrane or nitrocellulose membrane, and followed by blocking, and followed by incubation with the primary antibody, and followed by detection using the secondary antibody, and detection using the different detection techniques. There are different detection techniques depending upon the secondary antibody you use. Either it can be the chemiluminescence or a fluorescence or a colored based detection. So first we'll go to the first step. So first step is tissue preparation. So the samples can be taken from the whole tissue or a cell culture sample, or you can take the bacteria or a virus or environmental samples. Any source can be used to detect the proteins. And basically what you do is once you take the that cell or a tissue, you add a different buffers to it, and this buffers has salts and detergents. So what this salts and detergent does is basically it ruptured the cell membrane or a cell wall. So as a result of which all the proteins, DNA and everything whatever present in the cell will come outside. Once it is come outside, basically it gets solubilized into that buffer. So this is how you basically use you do the tissue preparation for Western blotting. And always remember that the Western blotting, whenever you do the any southern, northern or Western blotting, you have to do it under the cold conditions, because this what happens is basically there are a lot of enzymes which can act on these proteins or DNA or RNA and degrade it before you detect it. So that's why you have to do all these procedures under the cold conditions. Then it is followed by Sds Polyacrylamide Gel Electrophoresis. So SDS is sodium dodecyl sulfate. So basically what you do is you'll take the protein of your interest, that protein solution which you have isolated in the first step, and then you add the loading buffer to it. So what happens is when you add the loading buffer, this loading buffer has basically SDS, that is sodium dodecyl sulfate. Sodium dodecyl sulfate is basically negatively charged, and what it does is basically it degrades or denatures the protein, and also it imparts a negative charge to the protein. Because this is very important, because when you wanted to separate the proteins using the gel electrophoresis, you want your protein size protein to get separated based upon the molecular weight, not on the basis of the not on the basis of the charge of the protein. Because if you see inside the cell there are lots of proteins and they have the different charges depending upon the percentage of positive, negative or neutral amino acids. Depending upon that mixture of positive, negative, and depending upon the folding of the protein, the charge will be exposed on the surface of the protein. So basically, you want to separate the protein basically on the purely depending upon the molecular weight, not on the charge. So that's why you use the SDS. So that is the role of SDS in this loading buffer. So basically, it imparts a negatively charged to the all the proteins. And then this SDS page involves the two phases. First one is a stacking gel, and the second one is a resolving gel. Stacking gel is basically what it does is basically it stacks all the protein, and then resolving gel, it resolves all the protein. It separates all the protein based upon the size, purely depending upon the size of the protein. The proteins get separated on the SDS page electrophoresis. And then basically this the principle of this SDS page electrophoresis is electrophoretic mobility of different proteins, because depending upon the size, the proteins will move in the electrophoretic this SDS page gel. So the larger size proteins will move slowly and the smaller size protein will move faster. So depending upon their size, these proteins will get separated on the SDS page. So once that SDS page is done, so what you do is basically you transfer the proteins which are present on the SDS page onto the nitrocellulose membrane. So as I have mentioned earlier, nitrocellulose or a PVDF membrane, basically it has a charge. Basically, it has a charge and it has a pores. So because of which, and it has a high binding capacity, because of which the protein can bind to these membranes. And basically the same procedure as I have explained in the southern blot. So that is a technique, that is a same procedure which is used. Basically you place the gel, and on top of the gel you place the membrane and then filter paper, and then you basically you do the in kind of the in case of the electrotransfer, you apply the current, and then the proteins will move. The negative, all the proteins are negatively charged here because of SDS, and they move toward the positive charge. So it moves from down to up. So the membrane is present on the top, so that the protein will get transferred onto the membrane. And then after that you do the blocking. So basically once the protein get transferred onto the membrane, you have to block that membrane. So blocking is a method where, so because as I have mentioned earlier, this nitrocellulose has a charge, so it basically binds to the proteins, all the proteins. So that's why when you add the primary antibody, that primary antibody can also go and bind to the places where the protein is not bound. So it results in the kind of a non-specific detection of the proteins. To avoid that, the blocking is used. So in this blocking method, what you do is you use the protein, you use a dilute solution of protein. In this case, either it will be bovine serum albumin or non-fat milk. So this is used to block the spaces where the protein is not bound onto the membrane. So it basically it prevents the non-specific binding of the primary antibody. So after blocking, so what you do is you have to wash it. So basically in detection technique, what you do is after blocking, what you do is basically you add the primary antibody. Primary antibody is the a protein which is raised against the protein of your interest. Let's say if you wanted to detect a protein X or a protein, let's say I wanted to detect some of the DNA binding protein. So then I will use the primary antibody which is raised against that particular protein, and then I add it to this membrane. I incubate it with this membrane, and then after that it is followed by washing. So in this washing step, what it does is basically it removes the non-specific binding. Basically you use some detergent. So this detergent, what it does is it basically removes the unbound primary antibody. Then after that, it is followed by secondary antibody. The secondary antibody is the antibody, basically it detects the primary antibody and it is conjugated with the either the horseradish peroxidase or alkaline phosphatase or different tags which can be used for visualization. And once the secondary antibody is added, so you incubate it for 1 hour or so at 4 degrees, and then you wash it with the second using the this unbound secondary antibody can be washed by using the washing buffer, and then further you expose this membrane to the chemiluminescent substrate. So what happens is basically the secondary antibody has this alkaline phosphatase or horseradish peroxidase. It oxidizes this chemiluminescent substrate, as a result of which the light will be emitted and it will get exposed onto the membrane, onto the X-ray, as a result of which that can be visualized. So this is how this Western blotting works. And as you can see here, so basically you take the protein of your interest and you separate the proteins. And after that, what you do is basically you do the incubation part. So where you this is your membrane with protein, and you add the primary antibody, and then followed by secondary antibody with the specific tag, and then you add the substrate. And once you add the substrate, this enzyme will degrade that substrate or oxidize that substrate, and it will release a signal. And that signal basically what it does is this signal can be visualized by using the autoradiography on the X-ray film. So this is about the Western blotting. And basically there are lots of applications of this Western blotting. So one best example is it's basically used in medical diagnosis to detect the different antibodies against a bacteria or a virus in the serum. For example, if you want to detect the HIV, so what you do is you basically take the serum from the HIV infected patient, and then you do the either the Western blotting or ELISA to detect the whether the person is exposed to the HIV or not, or infected with the HIV or not. And this is useful for detection of the different proteins. For example, in kind of a prion diseases, where you can see whether these specific prion proteins are present in the person's serum or not. And then it is also used to determine the size and amount of protein in a given sample. So these are some of the applications of this Western blotting. And thank you very much. So sorry once again for the delay, and thank you one and all.